Moving on to transport of gases. Now here both oxygen and carbon dioxide, they are transported via blood. Some amount of these gases, they dissolve in the plasma, and in our blood plasma there is 90 to 92% water, and these gases will dissolve in that plasma water, but maximum amount is transported in the bound state. So first of all, taking up the transport of oxygen. Now oxygen, since it is less soluble in water, so only 3% of oxygen is transported in the dissolved state, while remaining 97% of oxygen is transported bound to hemoglobin. And this binding certainly is a reversible binding, because in the tissues this oxyhemoglobin which is being formed has to be dissociated, molecular oxygen has to again become free so that it can diffuse out of the blood capillaries into the tissues. So here this HbO2 that we have mentioned here, this is oxyhemoglobin. So 97% of oxygen is transported as oxyhemoglobin. Now there are certain calculations we can do. 1 gram of hemoglobin, it transports 1.34 ml of oxygen. Hence 100 ml of blood will transport. Now we have to know how much hemoglobin is present in 100 ml of blood. That is about 14 to 16 grams. So on an average we can take 15 grams. So 100 ml of blood would be transporting 1.34 into 15, and this will give us around 20.1 ml of oxygen. So our 100 ml of blood would be transporting 20.1 ml of O2. However, this is 20.1 would be the value when all hemoglobin is saturated. That means there is 100% saturation. But in the physiological conditions, 100% saturation is a rare thing. So this value nearly equals 19.4 ml. So this is actually 19.4 ml. Now out of this 19.4 ml, that means our arteries are carrying every 100 ml of blood present in the artery is transporting 19.4 ml of oxygen. Now in the capillaries, some of this oxygen will diffuse into the tissues. So every 100 ml of blood delivers 5 ml of O2 to the tissues if the tissue is at rest. That means it is not highly metabolically active at that time. So this again we can mention over here, that is every 100 ml of blood delivers 5 ml of O2 to the tissues. And with this we can add the tissue which is at rest. If the tissue is highly active, like in case we are exercising, in that case instead of 5 ml this amount can go as high as 15 ml. So the tissue which is at vigorous exercise will be able to extract 15 ml of oxygen from the arterial blood. So in this case, as we can see that at rest, since 5 ml is delivered and near about the blood is carrying 20 ml, so 5 upon 20 if we calculate, so that will come out to be nearly 25%. So actually our tissues are grabbing just 25% of the oxygen from the blood. Remaining 75% of oxygen stays there in the blood as a reservoir. So in case the tissue starts becoming more active, So that reservoir is already there, so more oxygen can then diffuse into the tissue. Now next thing here, that is coming to this transport of oxygen, oxyhemoglobin that we discussed. Now this oxyhemoglobin is oxygen plus hemoglobin. Now hemoglobin, as we know, is contained inside the erythrocytes, that is our RBCs, and if we see the structure of the hemoglobin, this is having 4 polypeptide chains, and these 4 polypeptide chains are 2 alpha chains and 2 beta chains, and each of this chain is having a heme porphyrin. So that is being shown here in the center, and this is the heme porphyrin. This is a organic structure in which in the center there is one iron atom, and this iron is in the form of ferrous, Fe2+, which is there in the center. So oxygen binds with this central ion, and in a way we can see here that since hemoglobin is having 4 iron porphyrin, so each iron porphyrin is going to bind with one molecule of oxygen. So one hemoglobin, that means, is able to transport 4 molecules of oxygen in all. So when it is fully saturated, it will be carrying 4 molecules of oxygen. Now all these 4 molecules do not bind simultaneously. Rather, when there is no oxygen, the affinity for that oxygen is less. So once the first molecule of oxygen binds, the affinity for the next molecule increases. Then for the next, it will further increase till it is fully saturated. So with every binding of oxygen, the affinity for oxygen is increasing. And this kind of influence that O2 is exerting on the various subunits of hemoglobin is called the positive cooperativity. That is, the oxygen affinity of the other subunits are affected because of another subunit which has bound to oxygen. And the binding of oxygen to hemoglobin primarily depends on PO2. So if the partial pressure of oxygen is sufficiently high, which of course is at sea level, there is enough binding and enough oxyhemoglobin is going to be formed. But when we go on high altitudes, so what happens? The atmospheric pressure decreases, although there too oxygen is 21% by volume. So the amount of oxygen in the atmospheric air is same, but the partial pressure would be less. Like suppose at high altitude, the atmospheric pressure is 500 mm of mercury. So that partial pressure of oxygen you will be calculating 500 into 21%. So that means what has actually decreased is PO2. And since PO2 is less, there would be less binding, less oxyhemoglobin formed, and our tissues will be getting lesser oxygen. Now if we draw a curve, the relationship between partial pressure of oxygen and the amount of O2 bound to hemoglobin, this would be a sigmoidal curve, that is a S-shaped curve. Now let us take the curve between the partial pressure of oxygen, which is shown here on the x-axis, and here we have on the y-axis percent saturation of hemoglobin. Now such a curve, the sigmoidal curve that we have obtained here, this is called oxyhemoglobin dissociation curve. Now in this curve we can see the shape, it is sigmoidal, and initially in a sigmoidal curve there is a lag phase, that is the when hemoglobin is having low affinity. Now with binding of oxygen, affinity increases, so that's why there is a sharp rise, and when all the sites are occupied by oxygen, so there we have this saturation. So in this case, the saturation level would be when all hemoglobin molecules are saturated, that is 100% saturated. And this would be at the normal pH of the blood, which is 7.4, and our normal body temperature, that is 37 degrees Celsius. Now from this 100%, if we mark the 50%, that is when hemoglobin is 50% saturated. So what is the partial pressure of oxygen when 50% of our hemoglobin is saturated? So that is a value around 27 mm of mercury. And this we can very well see that we had discussed the partial pressures. So whether it is arterial blood or venous blood, the partial pressure of oxygen is always more than 27. So that means our blood is at least more than 50% saturated with hemoglobin. Now this phenomena or this property, this is called P50 value. So P50 value is that partial pressure at which our respiratory pigment is 50% saturated. So we can just define P50 over here. P50 value. So this is the partial pressure of oxygen at which the respiratory pigment, and in our case this respiratory pigment is hemoglobin, is 50% saturated. Now this simply indicates that if the P50 value is very low, so that means that pigment is having very high affinity for oxygen. So in other words, we can put this as that P50 is inversely proportional to affinity. So in living organisms, what kind of respiratory pigments are favored or evolution has selected them are those respiratory pigments which have a very low P50. So that means even if less amount of oxygen is available to us, then too the pigment will suitably bind with the oxygen. Now let us see the factors which are going to affect this P50 value. The P50 value can alter and how that will affect the affinity and what will happen to this curve. So that will be followed after this. The factors that affect the binding capacity of hemoglobin with oxygen. Now there are number of factors, we can list them out one by one and then we will discuss them. So first of all, the blood pH. We have studied that the normal pH is 7.4, but this pH can change depending on there is more of H ions, hydrogen ions in the blood or less of it. The partial pressure of carbon dioxide, so that means PCO2 levels, temperature, presence of a particular compound 2,3-BPG, that is bisphosphoglycerate. It can also be called DPG, that is diphosphoglycerate, and the composition of hemoglobin. So let us begin with hemoglobin composition. Now in this case, as we have studied that adult hemoglobin has 4 polypeptide chains, and that is why that adult hemoglobin can be just abbreviated as alpha 2 beta 2. 2 alpha chains, 2 beta chains. But there is one more type of hemoglobin that is present in our lifetime, and that is when the fetal life is present. So in this fetal life there are 2 alpha chains, but there are 2 gamma chains. So fetal hemoglobin, which is written as HbF, is alpha 2 gamma 2, because there is a difference in this chain. So that's why it is labeled differently. Now this HbF has higher affinity for oxygen than HbA, And when we say it has higher affinity, in other words, we can say that HbF has lower P50 value than HbA. So therefore, if we plot the graph, that is the oxyhemoglobin dissociation curve, so on this graph what we will see is that the graph for HbF would be towards the left of the sigmoidal curve or the graph of HbA. And this point is important because at the placenta, now the placenta functions as the fetal lung. The lungs of the fetus, they remain collapsed. All the exchange is taking place between the maternal and fetal blood at the placenta. And at the placenta, there is a barrier which prevents the mixing of the blood, but that barrier allows the exchange of gases. So what happens at this barrier? That from the maternal blood, oxygen will dissociate from HbA because now there is a pigment available on the other side of the barrier which is having more affinity for it, and that is HbF. So oxygen will move from HbA to HbF, and that's how the fetus will get oxygen. Now another factor, that is pH. Now O2 binding properties of hemoglobin are also influenced by the level of pH. Now as the blood passes through more active tissues like exercising muscle, it picks up acidic metabolites. Now acidic metabolites like lactic acid is being formed, more carbon dioxide is formed, and these being acidic, the blood pH falls. The hydrogen ions are increasing. When there is more acidic content, there will be more hydrogen ions, and these hydrogen ions bind to hemoglobin and decrease its affinity for oxygen. So that means if there is more acidic content in the blood, the affinity of oxygen for hemoglobin will decrease. And as a result, what is going to happen to the oxyhemoglobin dissociation curve? It is going to shift to the right. That will be just be seeing. And what does it mean? When the curve shifts to right, means that hemoglobin will release more oxygen to that tissue. So wherever the tissue is metabolically active, it is producing more carbon dioxide, and because of more carbon dioxide, there is acidic content being created because carbon dioxide combines readily with water to form carbonic acid, and this carbonic acid dissociates into H ions and bicarbonate ions. So whenever carbon dioxide levels are high, there will be acidic content created, and that will favor dissociation of hemoglobin from oxygen. And molecular oxygen, once it becomes free, it will come out of the RBCs, and from the blood it will go into the tissues. Now this phenomena has been called Bohr's effect. This was given by a physiologist, Dr. Christian Bohr, who gave this opinion that in the presence of high PCO2, the affinity of hemoglobin for oxygen decreases. So as a result, what happens is that hemoglobin starts unloading oxygen and it starts loading off carbon dioxide. Now the other factor that we can mention is this 2,3-BPG, that is bis-phosphoglyceric acid. Now this is a metabolite of glycolysis and it is produced in the RBCs. Now we have to again remember that RBCs carry out only anaerobic respiration because RBCs lack mitochondria where otherwise aerobic respiration would take place. So here when there is low partial pressure of oxygen, there would be more of this BPG produced, and BPG binds with hemoglobin. So it is a very strong anion and it competes with oxygen to bind with hemoglobin, and in the presence of high BPG, again this ODC, that is oxyhemoglobin dissociation curve, will be shifting to the right. That means oxygen will be unloaded from hemoglobin. Now another one, that is if we see the dissociation curve. Now this dissociation curve we had drawn earlier, which was at the normal pH, that is 7.4 and 37 degrees Celsius temperature. Now here we are showing the curve being shifted to the right. Now what are the conditions in which this curve will shift to the right? When there is increase in partial pressure of carbon dioxide, that is in the tissue. And when partial pressure of carbon dioxide has increased, there also is going to be increase in hydrogen ions. There is going to be increase in 2,3 BPG. And since the tissue is metabolically active, there would be also increase in temperature. So any of these factors will augment the release of oxygen from hemoglobin, or we can say there will be dissociation of oxyhemoglobin. So all of these factors, if these factors are reversed, this oxyhemoglobin dissociation curve will be shifting towards left, and these factors are causing the shifting towards right. So this was our original curve and this is shifting towards right. Or in other words, we can also say that in these conditions, the P50 value of hemoglobin has increased. That means the affinity has decreased. That means hemoglobin will require a higher partial pressure of oxygen to remain bound to the same amount of oxygen. But in the systemic capillaries, such high PO2 is not available, so rather oxyhemoglobin will unload that oxygen. Now let us see in case of what happens when there is hemoglobin F, that is fetal hemoglobin. So as we said, fetal hemoglobin will always be towards the left of maternal hemoglobin. So in this case, this graph in the middle, this one is for HbA. And this one is also for HbA in the conditions that we had just now mentioned, that is when the affinity is decreasing because of increase in carbon dioxide, H ions, BPG and temperature. While this graph here, which is towards the left, will always be for HbF, that is the fetal hemoglobin. So the graph for fetal hemoglobin is always towards the left of the maternal graph. Now let us take a question here. Which of the following has maximum affinity for oxygen? Now here we have HbA, that is adult hemoglobin, alpha 2, beta 2. HbF, that is fetal hemoglobin, alpha 2, gamma 2. Now HbS is sickle cell hemoglobin, and sickle cell anemia is a genetic disorder in which the hemoglobin with the beta chain is having some differences in the amino acid, resulting in the defective RBCs. And this one will be anemic condition, that means it is not able to bind with oxygen properly. And carbon dioxide, that's a gas, that's a different thing altogether. So we have to basically consider the three pigments HbA, HbF and HbS. So out of this, the one which is having maximum affinity, that is certainly HbF. It has very low P50 value. So here our correct answer is 2. Now let us take another question. So here, which of the following will not shift the oxyhemoglobin curve towards right? Increase in partial pressure of carbon dioxide, that certainly shifts the curve to the right. Increase in 2,3 BPG, this 2. Increase in pH. Now increase in pH would be that it is going towards alkaline pH. While when hydrogen ions increase, there will be decrease in pH. So this is the odd one out, while increase in temperature certainly shifts it to the right. So here the correct answer, or the odd one out, is the third one. So this is the correct answer. Now let us consider another pigment that we have, that is myoglobin, and how does it, what kind of difference it has from hemoglobin. Taking up myoglobin. Now myoglobin it holds oxygen as a reserve. Now where is this myoglobin present? So as the term myo denotes, it is present inside our muscles, and muscles also, it is majorly in the red muscles, and this one has one single polypeptide chain. So unlike hemoglobin, which is having 4 polypeptide chains, one myoglobin molecule will be having only one polypeptide chain, and with that polypeptide chain there is one heme porphyrin, that is iron porphyrin, attached with it. So that means one molecule of myoglobin will bind only with one molecule of oxygen. So we can say the binding ratio of myoglobin to hemoglobin is 1:4. Now this myoglobin, however, has a higher affinity for oxygen than hemoglobin. So again the purpose is that the blood vessels which are carrying blood to the muscles, so there again, just like in case of fetal blood we had seen, that oxygen will dissociate from hemoglobin and move towards the muscle to bind with myoglobin. So whichever pigment is having higher affinity will certainly attract oxygen towards itself. If we plot a graph for oxymyoglobin dissociation curve, it is going to be rectangular hyperbola. So this again would be a difference from that of hemoglobin. So we can mention over here that oxymyoglobin dissociation curve is rectangular hyperbola. Now let us compile all the dissociation curves together, that is for HbA, HbF and myoglobin in one single graph. So we will get a picture like this, where as you remember this one we had started with, this was for HbA. So this is for HbA and this is oxyhemoglobin dissociation curve when it shifts to the right when there is high PCO2, that is the one shown over here. And this one with green, this is for HbF. So for HbA as well as for HbF, they are sigmoidal. But if you see for myoglobin, so myoglobin is to the most left and this is the one which is hyperbolic. It is not sigmoidal, rather it is hyperbolic. So this is the graph which you will get for myoglobin. Now moving on to some other aspects, like all our tissues, as we know, they require oxygen. But if there is less oxygen available to the tissues, such a condition is called hypoxia. Hypo is less, oxia word comes for oxygen. So low oxygen tension in the tissues. Now this hypoxia can be due to, one is low partial pressure of oxygen, that we had mentioned when such a thing occurs, that is when a person goes on high altitudes. So such a condition will arise. So low PO2 will be at high altitudes. Then another condition is anemia. Now anemia is either the number of RBCs are lesser, or maybe RBC number is normal, but every RBC as much hemoglobin it should contain, that is less. So either low number of RBCs or low hemoglobin. So again oxygen carrying capacity would be less and tissues would be getting less amount of oxygen. So that's what is anemic hypoxia. Now third one is the ischemic hypoxia. Now ischemic hypoxia is when there is obstruction in the blood vessel. So that means here PO2 is normal, the blood is having sufficient amount of hemoglobin, but the blood cannot be easily able to pass through the artery because the artery is blocked, and that blockage could be because of like cholesterol deposition, or maybe there is some air bubble has been introduced inside, any such condition will be called ischemic condition. So ischemia is obstruction in the blood vessel. And lastly, no such condition is there, but still tissues are not able to get oxygen. So that could be because of histotoxicity. Now histo is tissue, and toxicity, that means of course some toxin or a poisonous substance present inside the tissues which are inhibiting the uptake of oxygen. And here the best example, like we have cyanide poisoning. Now cyanide, we have studied that it is an irreversible inhibitor of one very important enzyme, that is cytochrome oxidase. And because of that, now the tissue cells won't be able to take up oxygen. So these are different levels of hypoxia. Then there is another condition, or rather if the hemoglobin gets oxidized. And in the hemoglobin we had mentioned that the iron is in the form of ferrous. And when oxygen binds with it, it still doesn't get oxidized. It remains as ferrous because formation of oxyhemoglobin is oxygenation reaction, not oxidation reaction. But here somehow, due to strong oxidizing agents, that could be some pollutants, there could be some drugs, because of which this iron has been oxidized to ferric. If such a thing happens, then that is called methemoglobin. So that hemoglobin will be now called methemoglobin, and this one cannot bind with oxygen. And since it is not able to bind with oxygen, so certainly there would be lesser supply, and such a condition will be called methemoglobinemia. And if such a thing happens in a child, the body starts getting cyanosis. That means when the tissue do not get sufficient oxygen, they start turning blue, and that is why it is also called blue baby syndrome. So methemoglobinemia is also the reason for blue baby syndrome. Now next we move on to the transport of carbon dioxide. Now carbon dioxide is a metabolic waste, because in all the tissues where oxygen is being used for oxidation of food, carbon dioxide is produced as a waste. Now this carbon dioxide has to be removed from the body. It is highly soluble and readily diffuses through the cell membrane. So the way in which carbon dioxide is transported, since its solubility is more than that of oxygen, 7% of carbon dioxide is going to be transported in the dissolved state. So it is going to dissolve in the plasma water and the tissue fluid, and that is how it is going to be transported. 23% of it is going to combine with hemoglobin, and this is going to form a compound called carbaminohemoglobin. So 23% of the carbon dioxide is going to be transported in this form. Now this form, however, we have to again remember that when oxygen binds with hemoglobin, it is not with the globin chain but with iron porphyrin. But carbon dioxide is combining with the globin part, that is the protein part, with the amino group, and that's why that compound is called carbaminohemoglobin, which can be simply written in this manner. So 23 plus 7, that would be 30%. Remaining 70%, that is the major amount of carbon dioxide, is transported in the form of bicarbonates. And this carbon dioxide, which diffuses into the RBCs, it forms carbonic acid, that is H2CO3, because of the presence of a particular enzyme present over there, and that is carbonic anhydrase. Now this enzyme, this is a very important enzyme, and this combines carbon dioxide with water present inside the RBCs to form carbonic acid. Now this carbonic acid, it readily dissociates to form hydrogen ions and bicarbonate ions. Now hydrogen ions, if they increase inside the RBCs, there is going to be decrease in the pH, or we can say the internal environment of the RBC is going to be disturbed. Now most of the proteins, they have a buffering action, and within the RBCs the hemoglobin combines with the H ions and forms HHB, that is hemoglobinic acid, while this bicarbonate, it diffuses out of the RBCs and goes into the plasma. So in this case we can just show this equation here as H2CO3, as we mentioned, this dissociates as H plus and bicarbonate, and this H plus combines with hemoglobin to form HHB, that is called or named hemoglobinic acid. Now let us look at all these equations where they are occurring. So here we can see a RBC over here, in which carbon dioxide plus water, by the action of carbonic anhydrase, a very fast acting enzyme, there is formation of carbonic acid H2CO3, which soon dissociates as H plus and bicarbonate. Now here this bicarbonate, it diffuses out into the plasma, while so this bicarbonate from here, it is diffusing out, while chloride ions from the plasma come in exchange, and this phenomena has been called the chloride shift. The excess of bicarbonate, they leave the RBC, and in turn chloride which moves in, that is called chloride shift, and thus we can say that the chloride content of RBCs in the venous blood would be higher in comparison to that of the arterial blood. Now what happens when this blood reaches to the lungs, when it reaches to the alveoli? Now in the alveoli, the amount of oxygen, the PO2 of oxygen is higher. So oxygen readily combines with hemoglobin, forming oxyhemoglobin. And as oxyhemoglobin is formed, there is unloading of CO2. So there is a reverse phenomena than what we had seen with the Bohr's effect. Now according to Bohr's effect, what was there? That is, in the systemic capillaries, since there is more CO2, so there is unloading of O2. But in the pulmonary capillaries, there is unloading of CO2 and hemoglobin is binding with oxyhemoglobin. So as soon as oxyhemoglobin is formed, there is release of CO2. So this is what is called Haldane's effect. Now Haldane effect, that oxygenation of the blood in the lungs displaces carbon dioxide from hemoglobin, which increases the removal of CO2. Now this CO2, once it has become free, so it will reach into that molecular CO2 will be diffusing from the blood into the alveoli. So here formation of oxyhemoglobin is an important phenomena required for the dispelling of CO2. And again, just like in case of oxygen we had mentioned, here again we can mention that every 100 ml of blood delivers 4 ml of CO2 to the alveoli. So we can see the role of oxygen and carbon dioxide. Now sometimes a person may be inhaling carbon monoxide. Now, carbon monoxide is a highly toxic gas which is produced due to partial combustion of the fuel. Now, this carbon monoxide has a very high affinity for hemoglobin, nearly 250 times more than that of oxygen. And the early Greek and Roman people knew about it, so they used to execute the criminals by exposing them to carbon monoxide. Now, this carbon monoxide, it binds irreversibly with hemoglobin. That means this carbon monoxide with hemoglobin is going to form a compound COHB, which is named carboxyhemoglobin. And this carboxyhemoglobin is a stable compound. That means it is not going to dissociate. And once carboxyhemoglobin is formed, that means this hemoglobin cannot deliver oxygen. So, as a result, the person would be dying due to asphyxiation. Although little amount of carbon monoxide is not going to be that harmful until unless nearly 70% or more of the hemoglobin is bound to carbon monoxide and it has formed carboxyhemoglobin. And in the presence of carboxyhemoglobin, whichever amount of oxyhemoglobin is left out, that graph will be shifting towards left. That means it is not going to release oxygen to the tissues. So, it would be like a hypoxic condition only, as we had seen in anemic hypoxia. Only thing was, in anemic hypoxia there was less transport of oxygen due to low hemoglobin or RBCs. Here hemoglobin is not available to bind with oxygen. It has bound with carbon monoxide. Now, we can use these informations and solve a question over here. Calculate the amount of carbon dioxide delivered by 1 liter of blood to the alveoli. Now, we have just now seen that 100 ml of blood delivers 4 ml of CO2 to the alveoli. Now, here the question is, how much would be delivered by 1 liter? So, that means 1 liter of blood would be delivering 4 upon 100, and 1 liter is 1000 ml. So, we can just simply calculate and the answer would be 40 ml. So, here the correct answer is option 3. Now, this brings us to the end of today's class, where we have discussed very important topics regarding exchange of gases and the transport of gases. In the next class, we would be discussing the mechanism how the entire process of breathing is controlled in our body. So, that's all for now. Thanks for watching. Have a wonderful day.